Hello, welcome to Biotechnology. The topic that we are going to discuss today is about antigen processing and presentation. The major antigen processing cells are macrophages, B cells, and dendritic cells. The macrophages is associated with MHC I, and B cells and dendritic cells will be associated with MHC II. Antigen processing is the process of proteolytic cleavage of protein by enzymes into small fragments and their association with MHC molecules by the antigen-presenting cells. This is an active process requiring energy. Antigen presentation is the presentation of processed peptides, in association with MHC molecules or PMHC on the surface of processing cells. There are two pathways depends on the antigen. 1. Endogenous antigens are processed in cytosol or in secretory vesicles and presented on class I MHC molecules to CD8 plus T-cells. 2. Exogenous antigens are processed in endosomes and presented on class II HC molecules to CR4 plus T-cells. 1. Endogenous antigens. Endogenous antigens are derived from proteins produced inside cell. These include altered self-protein antigens. Example: tumor antigens and non-self-protein antigens. Example: viral antigens. The endogenous antigens can be processed and presented by any nucleated cell. The pathway associated with endogenous antigen processing are: Step 1, processing of antigens into peptides. In this step, ubiquitin is covalently associated with antigen. This will target the protein to proteasome, which will cleave the antigenic protein to peptides and amino acids by proteases. Step 2, the Tap protein or transporters associated with antigen processing, which is present in the membrane of endoplasmic reticulum, in the form of heterodimer, TAP1 and TAP2, will facilitate the transport of peptides, from cytoplasm into lumen of endoplasmic reticulum. Step 3. Generation of class I MHC peptides. A chaperone protein called calnixin will bind to newly synthesized alpha chain of class I MHC and retains the class I MHC from being degraded until beta-2 microglobulin binds. A protein called tapasin will bind to the newly formed class I MHC complexes. It will form a bridge between the tap proteins and the class I MHC molecules. One more protein, calreticulin, will also bind to the class I MHC, in order to prevent lodging of any other peptide in agarrotype. Step 4. Association of peptides with MHC. Peptides replaces tapasin and calreticulin and bind to the agarrotype of class I MHC molecules to form PMHC. Peptide binding provides stability for class I MHC to allow transfer to surface. Step 5. Transport of PMHC to cell surface and presentation. The PMHC1 complex is transported from endoplasmic reticulum via Golgi bodies in a membrane-bound vesicles to the cell surface. The membrane of transport vesicles fuse with the cell membrane and PMHC complex, bind to membrane-presenting peptide lodged in agarrotype, toward exterior to be recognized by TC cell. Exogenous antigens are derived from proteins produced outside the cell. These include various bacterial, viral, protozoal, fungal and parasitic antigens which are derived from outside the body. Exogenous antigens associate with class II MHC molecules that activate helper CD4 plus T-cells for providing help to B and TC cells. Steps in exogenous pathway are: Step 1. Generation of peptides. The peptides bound to MHC class II molecules are derived from engulfed pathogens. APCs internalize antigens by phagocytosis, by endocytosis, or both. The exogenous antigen is degraded into peptides within these endocytic vesicles. Acids present in the endocytic vesicles activate proteases that degrade proteins into fragments. These peptide fragments are to be loaded onto MHC class II molecules. Step 2. Generation of MHC class II molecules. Class II molecules consists of two transmembrane polypeptides, alpha and beta, and a third molecules, called invariant, are nestled in the groove they form, until the antigenic peptides are not transported. The two chains of MHC class II molecules, alpha and beta, are associated into the membrane of the endoplasmic reticulum. The invariant chain, that bind to the groove, will transport the MHC class II molecule from the Golgi apparatus to the endocytic compartments. The signals in the cytoplasmic tail of invariant chain will lead to proper sorting of MHC class II. In the endocytic compartments, invariant chain is cleaved to leave a peptide fragment called clip in the binding groove. This clip will prevent the premature binding of peptides to MHC class II molecules. Step 3. Class II MHC peptide loading. A non-classical MHC class II molecule, called MHC DM, removes clip from the peptide binding cleft and helps to load the antigenic peptide into the groove or agarrotype of nascent MHC class II molecules to form PMHC. Step 4. MHC peptide transport. PMHC is transported into a membrane-bound vesicle to the plasma membrane. The membrane of transport vesicle fuse with the cell membrane and PMHC complex bind to membrane and displayed at the cell surface. It is then presented to T helper cells with appropriate TCR and CD4 molecules. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon.